Good day, everyone. I am Irish Jean and Tingoy, and welcome to the Krebs Cycle. What is Krebs Cycle? The Krebs cycle or citric acid cycle is a series of enzyme catalyzed reaction occurring in the mitochondrial matrix, where acetyl-CoA is oxidized to form carbon dioxide and coenzymes are reduced, which generate ATP in the electron transport chain. Krebs cycle was named after Hans Krebs, who postulated the detailed cycle. He was awarded the Nobel Prize in 1953 for his contribution.
It is a series of 8-step processes, where the acetyl group of acetyl-CoA is oxidized to form two molecules of CO2 and in the process, 1 ATP is produced. Reduced high-energy compounds, NAD, and FON2 are also produced. Two molecules of acetyl-CoA are produced from each glucose molecule, so two turns of the Krebs cycle are required, which yields 4 CO2 6 NAD, 2 FON2, and 2 ATPs. Krebs cycle is a part of cellular respiration. Cellular respiration is a catabolic reaction taking place in the cells. It is a biochemical process by which nutrients are broken down to release energy, which gets stored in the form of ATP, and waste products are released. In aerobic respiration, oxygen is required. Cellular respiration is a 4-stage process. In the process, glucose is oxidized to carbon dioxide and oxygen is reduced to water. The energy released in the process is stored in the form of ATPs. 36 to 38 ATPs are formed from each glucose molecule. Glycolysis, partial oxidation of a glucose molecule to form two molecules of pyruvate. This process takes place in the cytosol. Formation of acetyl-CoA, pyruvate formed in glycolysis enters the mitochondrial matrix. It undergoes oxidative decarboxylation to form two molecules of acetyl-CoA. The reaction is catalyzed by the pyruvate dehydrogenase enzyme. Krebs cycle, TCA or citric acid cycle, it is the common pathway for complete oxidation of carbohydrates, proteins, and lipids as they are metabolized to acetyl coenzyme A or other intermediates of the cycle. The acetyl-CoA produced enters the tricarboxylic acid cycle or citric acid cycle. Glucose is fully oxidized in this process. The acetyl-CoA combines with oxaloacetate, 4C, to form citrate, 6C. In this process, two molecules of CO2 are released and oxaloacetate is recycled. Energy is stored in ATP and other high-energy compounds like NAD and FOD2. Electron transport system and oxidative phosphorylation, ATP is generated when electrons are transferred from the energy-rich molecules like NAD and FOD2. Produced in glycolysis, citric acid cycle, and fatty acid oxidation to molecular O2. By a series of electron carriers. O2 is reduced to H2O. It takes place in the inner membrane of mitochondria. It is an 8-step process. The Krebs cycle takes place in the matrix of mitochondria under aerobic conditions. Step 1. The first step is the condensation of acetyl-CoA with oxaloacetate, 4C, to form citrate, 6C, coenzyme A is released. The reaction is catalyzed by citrate synthase. Step 2. Citrate is turned to its isomer, isocitrate. The enzyme aconitase catalyzes this reaction. Step 3. Isocitrate undergoes dehydrogenation and decarboxylation to form alpha-ketoglutarate, 5C. A molecular of CO2 is released. Isocitrate dehydrogenase catalyzes the reaction. It is a NAD+ dependent enzyme. NAD+ is converted to NAD. Step 4. Alpha-ketoglutarate, 5C, undergoes oxidative decarboxylation to form succinyl-CoA, 4C. The reaction is catalyzed by the alpha-ketoglutarate dehydrogenase enzyme complex. One molecule of CO2 is released and NAD+ is converted to NAD. Step 5. Succinyl-CoA is converted to succinate by the enzyme succinyl-CoA synthetase. This is coupled with substrate-level phosphorylation of GDP to form converted to malate by the addition of 1H2O. The enzyme catalyzing this reaction is fumarase. Step 6. Step A. Succinate malate is dehydrogenated by the enzyme succinate, which combines with another molecule of acetyl-CoA and starts the new cycle. Hydrogens removed get transferred to NAD+ forming NAD. Malate dehydrogenase catalyzes the reaction.
Significance of Krebs cycle.
The Krebs cycle or citric acid cycle is the final pathway of oxidation of glucose, fats, and amino acids. Many animals are dependent on nutrients other than glucose as an energy source. Amino acids, metabolic fatty acids undergo beta oxidation and get converted to pyruvate and other intermediates of the Krebs cycle. It is the major cycle and get metabolized. For example, alanine is converted to pyruvate. Energy is produced after the complete oxidation of nutrients to oxaloacetate. It plays an important role in gluconeogenesis, lipogenesis, and interconversion of amino acids. Many intermediate compounds are used in the synthesis of amino acids, nucleotides, cytochromes, chlorophylls, etc. Vitamins play an important role in the citric acid cycle. Riboflavin, niacin, thiamine, and pantothenic acid are part of various enzymes cofactors, FAD, NAD, and coenzyme A. Regulation of the Krebs cycle depends on the supply of NAD+ and the utilization of ATP in physical and chemical work. The genetic defects of the Krebs cycle enzymes are associated with neural damage. As most of the processes occur in the liver to a significant extent, damage to liver cells has a lot of repercussions. Hyperammonemia occurs in liver diseases and leads to convulsions and coma. This is due to reduced ATP generation as a result of the withdrawal of alpha-ketoglutarate and the formation of glutamate.